There are two types of cell respiration: anaerobic cell respiration, where there's a lack of oxygen, and aerobic cell respiration, where oxygen is present. Anaerobic cell respiration takes place in anoxic conditions where there is a lack of oxygen. The syllabus wants you to know that in yeast, anaerobic cell respiration leads to ethanol and carbon dioxide being formed as products. In animals, anaerobic cell respiration leads to the production of lactic acid. In the presence of oxygen in aerobic cell respiration, pyruvate is formed from glycolysis, which undergoes link reaction. Thereafter, acetyl coenzyme A is formed in link reaction, which enters the Krebs cycle. Lastly, there's a reduced NAD and FAD produced in Krebs cycle deposit electrons in H+ ions at electron transport chain, creating a proton gradient that results in chemiosmosis, which is the synthesis of ATP.
Glycolysis occurs in the cytoplasm of the mitochondria. During glycolysis, glucose is broken down into two pyruvate molecules. First, the glucose molecule is phosphorylated. Phosphorylation is the addition of phosphate groups to a molecule. In glycolysis, this requires two ATP molecules. Phosphorylation makes molecules less stable, therefore more likely to react. The phosphorylation results in phosphorylated glucose. The phosphorylated glucose molecule undergoes lysis to form two 3-carbon molecules. These 3-carbon molecules are oxidized to remove phosphorus groups and form two pyruvate molecules. Next, the link reaction. The link reaction occurs in the cytoplasm of the mitochondria. Glycolysis and Krebs cycle are linked together by this reaction. The oxidative decarboxylation of pyruvate and coenzyme A forms an acetyl compound attached to coenzyme A and carbon dioxide. The reaction is a decarboxylation, since pyruvate molecules loses a carbon atom, and the reaction is oxidative, since the lost carbon atom is oxidized to form CO2. CO2 is a waste product of the link reaction.
The Krebs cycle occurs in the matrix of the mitochondria. It starts with acetyl coenzyme A from the link reaction, transferring an acetyl group to the 4-carbon molecule in the diagram. This adds 2 carbons to the 4-carbon molecule, resulting in a 6-carbon molecule. The 6-carbon molecule now undergoes oxidative decarboxylation, where 2 NAD+ molecules are oxidized to reduce NAD. These then go back to the electron transport chain to become NAD+ again. This then causes a CO2 molecule to be released as a waste product of cell respiration. The carbon chain is now a 5-carbon molecule because of the carbon loss in the CO2. The 5-carbon molecule undergoes the same process of oxidative decarboxylation again. The molecule is now a 4-carbon molecule. This 4-carbon molecule undergoes rearrangement. During this time, 2 ADP molecules undergo substrate-level phosphorylation to become ATP molecules. 2 FAD molecules are oxidized to reduce FAD and return to the electron transport chain to become FAD again. 2 NAD+ molecules are oxidized to reduce NAD and then go back to the electron transport chain to become NAD+ again. The 4-carbon molecule is now rearranged and the cycle starts again. The net yield of the Krebs cycle is 2 ATP molecules, 6 reduced NAD molecules, and 2 reduced FAD molecules. This is how cell respiration creates energy in the form of ATP.
The reduced electron carriers, NADH and FADH2, from the Krebs cycle reach the electron transport chain in the mitochondrial inner membrane. The electron carriers release electrons and hydrogen ions. The released electrons enter the electron transport chain, which consists of transmembrane proteins that act as proton pumps. As electrons move through the electron transport chain, they lose energy, which is used by proton pumps to pump hydrogen ions from the matrix to the intermembrane space. This causes an accumulation of hydrogen ions in the small intermembrane space, resulting in an electrochemical gradient. This gradient causes hydrogen ions to passively diffuse back into the matrix through ATP synthase in a process known as chemiosmosis. As the hydrogen ions diffuse back through ATP synthase, ADP is converted to ATP. Finally, oxygen acts as the final electron acceptor, removing the de-energized electrons to prevent the chain from becoming blocked. Oxygen also binds to the hydrogen ions to form water in order to maintain the gradient to ensure that chemiosmosis can continue.